Meiosis is a complex process of cell division. The cell divides twice in meiosis, with 4 steps before each split. On the first split of meiosis, the 4 steps are known as prophase, metaphase, anaphase, and telophase, or PMAT for short. On the second split, the 4 stages are known as prophase 2, metaphase 2, anaphase 2, and telophase 2, as it is the second split of meiosis. Before the process of meiosis, the cell is in a stage known as interphase. This is where the cell can grow, replicate its DNA, and do its daily cell functions. Prophase 1 is the first stage of meiosis. In this stage, the following occurs. The nuclear envelope disappears, the centrioles start to move to opposite ends of the cell, the chromosomes start to condense, and the chromosomes match up with their homologous pairs. This is where crossing over begins. Crossing over is the exchange of genes between the 2 chromosomes. The second stage of meiosis is known as metaphase 1. Here, the chromosomes that have paired up with their homologous chromosome line up at the middle of the cell. These pairs are known as the tetrads. Spindle fibers attach to the centromeres of the tetrads. Anaphase 1 is the stage where the cell is starting to split. Here, the chromatids are pulled to opposite poles of the cell by the spindle fibers. Telophase 1 is the 4th and last stage of meiosis 1. This is a stage before the cell splits. During this stage, 2 new nuclei form around the 2 chromatid poles. Cytokinesis is not a part of meiosis 1, but is the stage where the cell splits into 2 new cells. Interkinesis is the next step. This is yet another step that is not part of meiosis, but is a step where the cell can rest, grow, and prepare itself for the next split in meiosis 2. The cell is now ready to move on to meiosis 2. Prophase 2 is the first stage of meiosis 2. During this stage, the chromosomes condense, the spindle fibers form, and the nuclear envelopes disappear. Metaphase 2 is the next stage of meiosis 2. Here, the spindle fibers from the centrioles attach to the centromeres of the chromosomes. Anaphase is the third stage of meiosis 2. This is a stage where the cell is preparing to split. Here, the chromatids are pulled to opposite poles by the spindle fibers. Telophase 2 is the last stage of meiosis 2. This is the stage before the cell splits into 2 new cells. In each cell, there is a new nuclear envelope that surrounds the chromatids. Meiosis 2 is now complete, though there is still one more step to go. Cytokinesis. This is not a part of meiosis 2, but is the stage where the 2 cells split into 4 cells. The long and complex process of meiosis is now complete. The 4 new haploid cells can repeat that process over once they have finished interphase. Mitosis is a process of cell replication. The cell replicates by duplicating itself. There are 4 steps to the process of mitosis: prophase, metaphase, anaphase, and telophase. There is 1 step before the process of mitosis. This is known as interphase. During this stage, the cell can grow, replicate its DNA, and carry out its daily cell functions, just like in meiosis. Prophase. Prophase is the first step of mitosis. During this stage, the chromosomes condense, the nuclear envelope disappears, and the centrioles start to move to opposite ends of the cell. However, crossing over does not occur, as the cell is basically doing a self-replication and does not require the trading of genes with another cell. Metaphase. During this stage, the chromosomes line up in the middle of the cell. The centrioles are now at opposite ends of the pole, which form spindles that attach to the chromosomes. Anaphase. This is the stage where the cell is preparing to split. Here, the chromatids are separated to opposite poles by the spindle fibers. Telophase is the stage before the cell splits. In this stage, the cell is almost ready to split into 2 new cells. There is also 2 new nuclear envelopes that surround the chromatids in each cell. The process of mitosis is now complete, though there is still one more step to go: cytokinesis. This isn't a part of the process of mitosis, but is a step where the cell splits into two new cells. Here, there's two new centrioles in each cell. With a nuclear envelope and chromosomes, they can start the process of mitosis over again, once they have completed interphase.